Okay, right, so let's have a look at ultrafiltration. Right, so when we say ultrafiltration, we're talking about filtering small molecules. Okay, so filtering small molecules, i.e. you've got ions, urea, glucose, amino acids, and obviously you've got water. Right, so the only thing that will not be filtered here would be large molecules. So this is not filtered. So that would be plasma proteins. You've got blood cells. Okay, right, so here we've got the glomerulus, which is here. You've got the Bowman's capsule. You've got two arterioles here. So the smaller one is known as the efferent, and this is known as the afferent. Okay, so as you can see here, there is a difference in diameter. So one of the questions that I gave you in the homework that I set was what creates the hydrostatic pressure. Now the hydrostatic pressure is basically the pressure exerted by a fluid. Okay, so this hydrostatic pressure could be due to high blood pressure. Okay, so what I'm trying to say is the higher the blood pressure, that would mean the higher the hydrostatic pressure. Okay, now this hydrostatic pressure in the nephron is created by the differences in the afferent and the efferent diameter. Okay, so the differences in the diameter is what causes that pressure that will then force small molecules out of the glomerulus into the Bowman's capsule. So this would be ions, this would be urea, this would be glucose, amino acids, and you've got H2O. Okay, so the only thing that will remain inside the glomerulus would be things like proteins. Okay, why is it that they can't? So you've got plasma proteins, sorry, so plasma proteins, and you've got blood cells. Okay, so like I said, the only reason why that these are not filtered is because they're too large. Okay, so when we say filter, it's almost like this glomerulus here is almost like a sieve. You know how you've got little holes in a sieve that allows, you know, small, like the water and, you know, small molecules to pass through, and then obviously filtering large ones. This is exactly the same as that. So it's filtering small ions, urea, glucose, amino acids, but stopping the movement of plasma proteins and blood cells into the Bowman's capsule. Okay, so one of the things that they could give you in the exam is basically to sort of explain why someone would have plasma proteins or blood cells. So if they have plasma proteins in the urine, so this is a good reason to suspect that they could have kidney failure. Okay, right, another thing that they will expect you to know is how are these molecules passing from the glomerulus into the Bowman's capsule. So basically, they passes through the endothelium, then they go straight to the basement membrane, and then goes through the podocytes. So the podocytes, if you just look at the file that I sent you, the podocytes are basically found in the Bowman's capsule. Okay, so these are the special cells found in this area here that will allow the filter to pass through. Okay, so I've just covered the ultrafiltration, so we will have a recap of this when I come back next week. But as far as this is, this topic is about, so you just need to talk about what is ultrafiltration. So when we say ultra, we mean small. Okay, and what are we filtering? We're filtering these. Okay, and the filtrate. So this basically will form the glomerular filtrate. Okay, so I've paused the video now. Then what I'll do is I'll send another video to explain selective reabsorption and then osmoregulation. Okay, so you can watch this video before I come back next week, and then we can go over again. I mean, ultrafiltration and selective reabsorption are pretty easy. It's the osmoregulation that will probably take me a long time.